My senior research project was investigating the impact that probiotics would have on reestablishing the gut microbiome following an antibiotic treatment. The microbiome is a collection of viruses, bacteria, and fungi that interact with the structures around them. Some of these microbes are responsible for creating an inflammatory response, while others directly impact organs by protecting their epithelial lining. Most of the time, bacteria live symbiotically within their host, but opportunistic pathogens can take advantage of a disrupted microbiome and colonize within the gut. When these pathogens take over, they can cause deadly diseases like Clostridium difficile infection, necrotizing endocarditis, irritable bowel syndrome, and Crohn's disease. What we were looking for during this project was how large of a dysbiosis or a disruption would occur from an antibiotic treatment and how probiotics could help solve that problem. So to do this, we would use rats due to their internal similarity to humans. Probiotics are species of live bacteria that are taken as supplements with the goal to restore healthy symbiotic bacteria into the gut. This study focused on using a Lactobacillus species as our probiotic. Previous studies have shown that they have had a positive impact on the gut microbiome of many different mammals, including humans. So we started out with L. brevis and L. rhamnosus and decided which bacteria would work the best for us. Eventually, after culturing and plating both on MRS, we realized that L. rhamnosus grew the most effectively, and we moved forward with that for our project. L. rhamnosus is a gram-positive rod-shaped bacterium and is found abundantly in the gut of healthy organisms. Antibiotics are antimicrobial substances used to fight off bacterial infections. As chemotherapeutic agents, antibiotics either kill or inhibit the growth of bacteria that is capable of causing infections in the host. There are multiple mechanisms to killing bacteria. The vancomycin inhibits the formation of the peptidoglycan layer, which in turn does not allow for the formation of the bacteria's cell wall. Vancomycin specifically inhibits the penicillin binding proteins, which makes the cross-links within the peptidoglycan layer. Antibiotics can kill pathogenic bacteria, but they can also kill the healthy native microbes that are within the gut microbiome. So the misuse of antibiotics can lead to antibiotic resistances, where the bacteria evolves to withstand these treatments, and therefore, the more an antibiotic is used, the more destruction it can cause to the host organism and their gut microbiome. So what I was investigating was to see if the probiotic L. rhamnosus would be capable of restoring the gut microbiome of rats following the depletion of bacteria within their gut caused by an antibiotic treatment of vancomycin. I then hypothesized that L. rhamnosus would be able to restore the gut microbiome of rats following this antibiotic treatment that depletes their gut microbiome. Throughout the study, I had 16 male Sprague Dawley rats. I had 4 groups, where the first group was a control group with no antibiotics and no probiotics, a second group with no antibiotics but with probiotics, a third control group with antibiotics but no probiotics, and a fourth experimental group with both antibiotics and probiotics. Daily, the rats were weighed, fed, and given water, and eventually this would include giving them their carrot juice that would either have probiotic, saline, or the vancomycin treatment within it. The rats were randomly placed into cages based off of their initial weights when we first received them. The administration of supplements occurred for a total of 14 days. Our rats treated with antibiotics were intraperitoneally injected for the first day of treatment, and then for the next 2 days of the 3-day treatment, the vancomycin was administered through their carrot juice. The vancomycin we had was at a concentration of 20 milligrams per milliliter, and the volume injected into each rat was based off of the rat's weight, which our injections ranged from 0.24 milliliters to 0.27. For our oral administration, we gave 4 milliliters of vancomycin and added that to 21 milliliters of PBS and 25 milliliters of carrot juice, which would give us a total of 50 milliliters within each of their bottles. So after the 3-day vancomycin treatment, we were then able to start our probiotic supplement groups. So we created L. rhamnosus pellets by centrifuging 5 milliliters of culture at 14,000 RPMs for 17 minutes. That gave us the pellet. And then we inserted the pellet into 25 milliliters of carrot juice and 24 milliliters of PBS to again make a total of 50 milliliters within the probiotic treated bottles. The rats that were given the antibiotics initially or no treatment at all were given 25 milliliters of PBS and 25 milliliters of the carrot juice following the initial antibiotic treatment, and this continued for 11 more days. Before the rats were euthanized by CO2 asphyxiation, they were weighed one more time. With this data, we could then determine the weight gain each group experienced through the experimental process of injection day to euthanization day. So this table here summarizes the weight gain that each group experienced, as well as the percent change in weight. The rats treated with antibiotics and probiotics had the largest overall weight gain at 90.635 grams throughout the experimental process, and they also had the largest average weight gain per day at 6.47 grams per day. However, there was no one treatment group that had any recognizably larger weight gain than any other. As you can see, their change in weight percentage was all pretty similar, ranging from 27% to 35%. All of the dissections were performed directly after the rats were euthanized, and these were done by my lab partner, Alexis Mosier, while I was completing the microdilution plating simultaneously. The cecum from each rat was weighed, measured, and recorded during this dissection, and this graph shows our data that we obtained from that. So this graph is comparing the cecum weight in grams versus the cecum length in centimeters. Each point on this plot represents a single rat cecum and is color-coded by group. So the control animals with no probiotics and no antibiotics are represented with the blue color. The animals receiving probiotics but no antibiotics are in red. Animals receiving antibiotics but no probiotics are in green. And animals receiving antibiotics with probiotics are in purple. So with this, we obtained a correlation coefficient R that was equal to 0.7138 and an R-squared value equal to 0.5096 that also had a positive slope. So this is saying that there is a positive correlation of an average strength between the cecum weight and the length, so that when one increases, so does the other. A correlation was also tested against the cecum weight and the concentration of bacteria found within each of the cecum. So this gave us a coefficient value of 0.503 and an R-squared value of 0.253. So these calculated values are weak since they are closer to 0, but they are nonetheless still positive, showing that there is a weak correlation between the total bacteria concentration and the cecum weight. So again, this is showing that when the cecum weight increases, that the bacteria concentration in that cecum also increases as well. Microdilution platings were done during the dissection of the animals so that their cecum could be swabbed and placed into a PBS solution. This PBS solution with our swab in it was known as our 10 to the negative first dilution, and from there we took it down to 10 to the negative eighth by taking out 100 microliters from each sample and putting it into the next tube. This then would be able to finalize our 1 to 10 dilution scheme. After that, we took 4 10 microliter dots of each sample that were placed onto their corresponding area on a labeled quadrant of our MRS plates, like can be seen on this slide. After that, we incubated them at 37 degrees Celsius overnight, and then pulled them out and were able to determine what type of colonies and counted the colonies that grew on them. So what we found was 2 different colonies grew. Colony 1 was yellow, circular, and slightly raised, while colony 2 was white, circular, and blotchy with undefined borders. And then after counting all of the colonies within those 4 little circles on the picture on the right, we were able to calculate their CFU per milliliter. So now that all the colonies were counted off of the plates that were shown in the previous slide, we were able to calculate the colony forming units per milliliter, or the CFU per milliliter. We took the average of these calculations that we found from each treatment group and put this into the bar graph for both colony 1 and colony 2. So colony 1 is represented with the striped bars, while colony 2 is represented with the checkered bars. I performed a statistical test, a two-way ANOVA, which revealed that there was no significant difference within the treatment groups with a p-value of 0.2965, but there was a significant difference between colony 1 and colony 2 with a p-value of 0.0015. We found that colony 1 had a significantly higher concentration in each of the treatment groups than colony 2 did. So contrary to my hypothesis that the probiotics would be able to resupplement the gut microbiome. The antibiotic treated rats actually had the highest number of colonies forming in their cecum. So colony 1 was found at an average of 5.08 times 10 to the 8th CFU per milliliter, and colony 2 was at an average of 1.33 times 10 to the 8th CFU per milliliter in the antibiotic treated rats. This would give us a total of 5.46 times 10 to the 8th CFU per milliliter, while the smallest concentration of bacteria was found in the probiotic only treated groups, and they had an average of 2.21 times 10 to the 8th CFU per milliliter. We also performed molecular work by extracting the DNA from the rat ceca, and we had unsuccessful results with our PCR. So if we would have saw results from that, our findings may have been a little more conclusive as to why this occurred. So overall, this study did not show any positive support that probiotics can be used to reestablish a depleted gut microbiome following an antibiotic treatment. However, since the antibiotic treated groups had the highest concentration of bacteria, the study really did not show that the vancomycin was capable of depleting the gut microbiome. The higher concentration in the antibiotic treated groups could have been due to the vancomycin killing a certain bacterium and allowing more room for other bacteria to flourish within the gut of the rat. The probiotic treated groups may have had the lowest concentration of bacteria because by implementing more bacteria through the probiotic pill, it could have taken up more nutrients in the gut, and then the bacteria might have outcompeted each other and ended up killing more bacteria within the probiotic treated groups, giving it a lower overall concentration. The oral administration of the probiotics and the antibiotics on the second and third day gives a lot of room for error, even though that the rats never had more than 20 milliliters of their carrot juice remaining. This still could be enough for the probiotic and or the antibiotic group to not be effective in their treatment. I would like to acknowledge the A.J. Palumbo Foundation and the St. Vincent College Biology Department for funding and supplying my research project. I would like to acknowledge my research advisor, Dr. Jennifer Koehl, for being helpful, supportive, and dedicated to my research project. My collaborator, Alexis Mosier, my fellow senior Koehl staff member, Caitlin Akenclose, and our research assistant, Colby Hannon, as well as all of the junior Koehl staff members that helped out with our project.